Place 370 gives us a lot of lessons to be learned, because everyone was excited about the design of the study based on pretty limited initial data. Phase 1b study, which looked at the combination of cobimetinib, atezolizumab, a MEK inhibitor plus a PD-L1 inhibitor in a later line setting in about 20 patients with MSS. I mean, most of them had MSS tumors. And we saw 4 responses. 3 of those were really MSS tumors. Once we didn't know what the MSI status was. And this created a lot of excitement initially. Everyone wanted to make immunotherapy work in these otherwise immune therapy refractory colorectal cancers. And there was some preclinical rationale. There was the idea of recruiting T cells into the tumor through the MEK inhibition, making these T cells be more sustainable, have durable activity, etc. So there were a lot of data that kind of led to the idea of, let's run a phase 3 study. Now, the phase 3 study eventually accrued very rapidly. It was an international effort and randomized patients to atezolizumab, cobimetinib versus atezo alone versus regorafenib as the control arm, which was used because it was considered, an easy arm to beat based on toxicity profile and limited activity. So the study eventually showed, it came out negative. There was no difference in outcome between either the combination arm atezo cobimetinib or atezo alone compared to regorafenib. In fact, regorafenib outperformed expectations and now being cited for regorafenib being actually associated with overall survival about 8.5 months in a later line setting, in a phase 3 setting. So I think that's encouraging to know we have an active agent now, even in modern times, that held its ground in a phase 3 setting. Now, the lessons learned are several. First of all, we moved very rapidly from limited data in phase 1b to expanding into a phase 3 study. And we need to be very careful for future endeavors, really probably to have better signals. I mean, this is also important right now for the excitement people have based on some limited data on regorafenib plus nivolumab. The data from Japan in gastric cancer and colorectal cancer. Are those enough to really warrant a phase 3 study, in particular when now, single arm studies, from other institutions do not seem to be able to completely replicate these data. And I think it's a very important point to be careful here in the development to see what are the patients that could benefit, for instance, from this combination. This other part is, cobimetinib atezolizumab not only did not beat regorafenib, but was also more toxic. So a lot more patients, 2 times more patients went off therapy with cobimetinib atezolizumab compared to rego due to side effects. So it's not just this drug didn't work, we might have also potentially harmed some patients, at least with side effects. So all this is important to really have good data that allows us to move combination therapy, immune combinations into a later line setting, into phase 3. I know people are more careful now. On the other hand, everyone recognizes if we can make MSS tumors, MSS colorectal cancer immunogenic, that'd be it's a home run. It's a holy grail, of immune therapy, because it's a very common and very immune therapy refractory cancer. So we all want to make sure that we find eventually, and I'm sure we will, find ways to treat MSS cancers with immunotherapy.
What we have seen, there are several possibilities in metastatic colorectal cancer to try to enhance the immune response in the vast majority of metastatic colorectal cancer patients whose tumors are microsatellite stable. And these tumors, we know, could be either immune desert or immune inflamed. And both conditions are not the best conditions for the conventionally IO like anti-PD-1 or anti-PD-L1 antibodies to work. In the past, we had some hope that by inhibiting MEK pathway, we could enhance in these patients the activity of PD-L1 antibodies, but this strategy unfortunately failed. What has been now tried to be done is a dual approach. One is in RAS wild-type patients, try to combine anti-GFR monoclonal antibodies, and most likely to combine cetuximab, also because is an ADCC potential immunoglobulin inducer with anti-PD-1 or anti-PD-L1 antibodies. And there are some studies that are currently ongoing ongoing also in colorectal cancer. Some of them are ongoing with these combinations in head and neck squamous cell carcinoma. And some attempts have been done also in non-small cell lung cancer. Or another opportunity has been to try to combine immuno-oncology drugs such as PD-1 or PD-L1 inhibitors with anti-angiogenic drugs. For example, we know that the combination of small molecules that block angiogenesis, such as the ones that are used in first-line renal cancer, combined with anti-PD-1 or anti-PD-L1 antibodies, such as pembrolizumab or avelumab, respectively, have become the standard therapy for these patients. We know, for example, that combining bevacizumab and atezolizumab, it is another PD-L1 antibody, is becoming the first-line potential preferred therapy for hepatocellular carcinoma. And so there is a strong rationale of combining either anti-VEGF A monoclonal antibodies or pan-tyrosine kinase anti-angiogenic small molecules with PD-1 or PD-L1 antibodies also in metastatic colorectal cancer. We had a report last year of interesting data in MSS patients with refractory disease of combining regorafenib and nivolumab. At the current annual Congress of ASCO, some data have been presented in an abstract and then in a poster presentation on the combination of regorafenib with the PD-L1 antibody, avelumab. The rationale behind this type of combinations is that this broad spectrum of action of regorafenib, blocking several tyrosine kinases that are important for endothelial cells as well as several cells that are involved in the microenvironment in the tumor to, in some way, interact with the T effector cells. This inhibition could increase the activity of PD-1 or PD-L1 antibodies. And I hope we will have more data in the near future, because unfortunately, the bottom line is that whereas, especially after the Keynote 177 randomized phase 3 trial, we know that in MSI patients, pembrolizumab, and therefore a PD-L1 antibody, is very effective in these patients. However, these patients are only 5% of metastatic colorectal cancer patients. So what can we offer in terms of immunotherapy to the other 95%? This is a very important open question in which the combination with other molecular target agents such as EGFR inhibitors in selected patient population, RAS and RAF type, or with broad spectrum anti-angiogenic drugs such as regorafenib, is very interesting and worth to be addressed in clinical studies.